Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, patient advocate Donna Cryer, liver wellness advocate Louise Campbell, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on the Surfing the NASH Tsunami podcast.
For everyone with an interest in NASH, or more broadly fatty liver disease, Surf's Up. Season 2, Episode 5 of Surfing the NASH Tsunami starts now. We have just finished attending the FDA's webcast titled Regulatory Perspectives for Development of Drugs for the Treatment of NASH. This widely anticipated event was hoped to provide guidance and insight in some areas. I would say it raised more questions than it answered, but we'll talk about that as we go through this discussion. I'm going to forego all the topics I usually discuss when starting the podcast so we can dive right into today's content. Our participants include the three regulars, Stephen, Louise, myself, plus Professor Manal Abdelmalek of Duke, who was with us at university, who was with us on Monday. And we're also joined by Dr. Naim Alkhouri of Arizona Liver Institute, no stranger to people who listen to the podcast. He's been with us several times before. Welcome, Naim. How are you today?
I'm good, Roger. Thank you for having me.
Always a pleasure, and I'm glad you could make it. And also, we're joined by Kitty Yale, the Chief Development Officer at Akero Therapeutics. If you listen to the podcast we taped on Monday, you heard on Thursday, Kitty was good enough to share some specific thoughts that we included in the pre-event episode. Kitty, welcome. Good to have you here.
Yeah, glad to be here, Roger. Thank you very much for inviting me.
Oh, our pleasure. Please, if you could, take a couple of minutes and tell our listeners a little bit about your role today, your career to date, and please be certain to share at least one fact that we wouldn't know if you didn't tell us.
I'm the Chief Development Officer at Akero Therapeutics. We're a small, clinically focused startup biotech company. I've been there for 2 years, and really my role there is focused on the clinical development of efruxifermin, which is a FGF21 analog. Prior to Akero, I was at Gilead for 17 years. Very fortunate to be focused on working in liver disease for many years. And, at Gilead, it was especially important as we were sort of very focused with sofosbuvir on HCV clinical development, but then later on sort of was involved with the Gilead early NASH studies. In terms of things that people don't know, I think Stephen knows, but my accent's a little strange. I've been in California for 20 years, but I'm originally from Scotland. And I suppose the one thing that maybe is unusual about me for a biotech individual, I'm the fourth daughter of a farmer who needed a son. But it is amazing. I've managed, Stephen and I like to tell early stories about growing up on a farm. So it has been actually useful for my overall drug development.
So can you give us one very brief snippet of a story that demonstrates that it's been useful?
Stephen and I had some interesting stories talked about dealing with vermin on farms, but I don't think I can talk about it more publicly because Stephen's stories, they have more snakes in southern US than they have in Scotland. So I'll just say it might frighten off the listeners.
Well, then that's a fantastic introduction, and thank you for not scaring anybody away this early in the discussion. With that, let's just get going. I want to ask each panelist 2 questions about today's webcast. Number 1, your general feeling about the event on a 0 to 100 scale, where 0 equals left me completely cold and 100 equals left me red hot with excitement. And the second is one particular statement or answer to a question, or if this is the case, non-answer to a question in this meeting that played a role in shaping your reaction and the number that you gave. Brave one, go first.
Hey, Roger, this is Stephen. I'll go. So again, I think it's brilliant to have this podcast to discuss what was recently reviewed by the FDA. And I would give it a 5. So I'm not on fire with the discussion points and the answers, but I do think there were some fresh insights and actually some answers to questions that we had. The 2 that come to mind the biggest are, number 1, the clear guidance that digital slides could be used for interpretation and analysis of histopathologic surrogate endpoints. That's been a huge debate. It's been something that's been bantered back and forth. And I would say it's probably not the biggest fan favorite of pathologists. They do like to have the ability to kind of look in and out of phase, if you will, to look at potentially at more of a 3D image. And that's always been a pushback from pathologists. But understanding the speed at which we need to move to have these slides adjudicated, it's important to hear them say that they're accepting digital path, even for a phase 3 registrational trial. So that was important. Number 2, I thought it was really unique that the speaker that showed the slides, I can't pronounce his last name, showed that you don't have to do phase 4. You could actually do a well-compensated, simultaneous, parallel phase 3 study in cirrhotics, and at the same time do your standard F2, F3 NASH trial. You get approval, you hit your surrogate endpoint for F2, F3, you file your NDA, while your simultaneous, parallel, well-compensated F4 trial is ongoing, and you follow that out to adjudicate endpoints. And then that came back around as a question to Joe Turner. And the reason he defended that was because of the issues surrounding phase 4 in the setting of having an FDA-approved therapy. Would patients in placebo continue into the long-term follow-up phase? It's interesting now that potentially there is an avenue to acceptance without going through a phase 4 paradigm. So there's a lot more I could talk about, but those were two that really hit home to me.
I suspect we'll get back to them as we unpack the meeting a little bit, but thanks for kicking off, Stephen. Who would like to go next? Second brave one.
It's Manal, and I think I also was, around a 60% on the FDA. And I'll tell you, there's some things that I thought were informative. The FDA clearly wants to seek, a dialogue, partnership, is excited about this area of drug development. We're not anywhere close to histology going away. That is clear that that's still going to remain a surrogate. And what also resonated is really the onus of responsibility on us in academia and drug development and biotechnology to move forward with generation and validation of these biomarkers and implement them in the context of our clinical trials such that the FDA can deliberate on the strength of a surrogate to histology. And so I think we have a lot of work to do there, and that didn't really shed any new light or new position of the FDA. But I think we now have to put these validated biomarkers, FAST being one, for example, into practice and integration into our clinical trials. And I think where the meeting gave me a little bit of pause is the area which we have the most unmet need, our patients with cirrhosis, that we really, short of doing the registration trial after the NASH registration endpoint is met, we don't have any accelerated path for this cohort. We don't have any clearly defined non-clinical endpoints. And with that, we're still in a traditional approval pathway for cirrhosis. I think that's more of a challenge to the field than it is an issue with the FDA, but we really have to focus on this highest risk unmet need cohort.
Thanks, Manal. Second great answer. Who would like to go next?
This is Kitty. I think maybe my expectations were maybe a little moderate going in. I did hear Stephen's comment on the podcast in the beginning of the week that he was hoping that the presentation may really use the recent paper as a sort of starting point and go further. But I think I was sort of expecting the agency more to reiterate. And I think the presentations were somewhat introductory in some of their nature, but I did like the Q&A at the end. And I think that's where we got some of the more meaningful nuggets. So again, I was sort of like at 50%. I'm glad that Stephen picked up on the question about digital pathology. I actually asked that question, and I think it was very nice to get very clear guidance. And I think as we're getting very sort of guided towards multiple pathologists, being able to digitize that is a way forward. It was also interesting to see that they are open to actual artificial intelligence and somehow incorporating that into the pathology read, which I think will strengthen the overall read and will ultimately decrease variability. The second item I was paying a lot of attention to was also the area that Stephen was talking about really looking at how you could use two parallel studies and use the clinical outcomes from the cirrhotic F4 patient population and perhaps run that in parallel with your F2, F3, and then just take that through to a surrogate endpoint. I think what we'll need to get is real clarity that you're not expected to create separate outcome data for the F2, F3 population and whether that F4 would be acceptable for your entire package. So I'm curious what the others think and how they interpreted that conversation.
Naim, you want to go next?
Sure. I think maybe I'm more excited. I'll give it like a 70%. I feel like we do have a new endpoint in histology-based trials, phase 2b/3, which is achieving both NASH improvement and fibrosis regression, or NASH resolution and fibrosis improvement. I think this is going to help us to decrease the placebo effect. I don't know if you remember, Roger, at the liver meeting, Stephen presented data with aldafermin, for example, looking at achieving these two endpoints with NASH resolution, fibrosis improvement. If you look at the placebo response rate, that was at 0% in that small trial. So I think, the placebo effect has been derailing many of the trials, and I think having, an endpoint that has both the NASH endpoint and fibrosis will help us decrease the placebo effect. There was the data with Inventiva also, with lanifibranor, where they actually achieved both of them. And if you look at the placebo response rate in achieving both, it was about 7%. So definitely lower than what we've seen in other trials, with elafibranor, for example, where the whole trial was really jeopardized by the high placebo response rate in a way. So that was exciting. I think the other point, and we talked about it a little bit before we started the broadcast, was that if we're going to have this parallel trial design and having the F2, F3 not require any clinical endpoints, what is the sample size? I mean, this is a big deal. Can you actually design this with 600, 800 patients, get approval, and then wait on the cirrhosis trial to show outcomes? I think this will accelerate drug approval. We need more clarity on that point. I think this is a very important point. During the Q&A, I was a little bit disappointed with the fact that they will not really allow any trials based on cardiovascular outcomes or diabetes outcomes, because you still need the histology, so you cannot power the study to do this. I feel like this is a missed opportunity, but at this point, I don't see it happening anytime soon. It'd be nice to design a study in high-risk patients, maybe patients with type 2 diabetes, cardiovascular outcomes, include 6,000 people, follow them, look at overall mortality, cardiovascular disease, and liver outcomes. But this is not happening anytime soon.
Thanks, Naim. Excellent points all. Louise, this is the first time you ever heard one of these, so I'm intrigued by your reaction.
Yes. At one stage, I just thought they were reading out from the documents rather than actually putting the discussion forward. But as everybody's commented, that improved, and certainly the questions and answers shed more light onto the thinking. And I think if I was to rate it from my perspective, I'd probably go about 60%, because I think, as Manal and the others have detailed, there is now an ability to use more surrogates, and an improvement in the surrogate can be taken as an improvement in health, which I think is a step forward. I think we do have to really use the surrogates quite accurately if we're going to do proof of concept in the early phase studies, because it doesn't take too much to skew a FibroScan. You can do that very easily. It can happen in different of the other markers as well, I think. So therefore, to be able to concentrate to make sure that our sort of biomarkers and non-invasive techniques come together, they're patient-friendly. If we can prove them in the clinical trial world, it's probably where we're going to be having the strengths after the trial and the implementation into real-life situations. And I think for countries that don't have the wealth of medical techniques and specialists that the US have in Europe and some of the big centres in Asia, I think going to non-invasive and biomarkers, if we can really strengthen their use within clinical studies, and Manal mentioned FAST there, they do have weaknesses. But if you can prove benefits even on the weaknesses, then the strengths are there and they'll be even more highlighted. So I think that for me was interesting. I think it allows us to move beyond the clinical trial environment into real-world application a little bit easier than just using histopathology, I suppose. But I did like, as Stephen commented, and Kitty, the fact of the AI being advocated, and I think that will be helpful to pathologists and histopathologists. We'll start to trust it a little bit more if it's unified. So I enjoyed it overall. Some areas were as clear as mud, but other areas I thought were very nicely clarified.
Thanks, Louise. I'll go last. They teach you in business school that the correct answer to every question is it depends. So I'm not sure whether in my mind this is about a 55 or about a 70. And the question on which my uncertainty rests is something that Kitty and I both picked out of the document that we talked about on Monday, but didn't really come up today, which is when they start talking about the level at which they need to scrutinize for side effects and what does that mean in terms of overall sample size. The ICH document would say 1,500. That sounds like a lot of patients and aligns with things like, can you get all this done in one trial? I was hoping that there would be a little more guidance on how to manage the safety side of having enough patients to measure, to see what they wanted to find. And that wasn't addressed. Now, the things that were addressed are probably more important and more immediate. And there, I think the answers were quite possibly a 70, unless the answer to this one is really crippling. But I just don't think we know yet. I think we're going to have to figure that out. With that, we have about 1 hour left to dive into the meeting itself. In the informational brochure, the FDA promised to discuss 5 topics, which they did in fact discuss. Phase 2 drug development in NASH, clinical trial inclusion and exclusion for Phase 3 NASH trials, surrogate efficacy endpoints that have been acknowledged by FDA as reasonably likely to predict clinical benefit, clinical endpoints for the confirmation of clinical benefit, and strategies of clinical drug development for NASH. I think given the time that we have, it might be reasonable not to discuss Phase 2, Phase 3, and cirrhosis separately, but to go to the other 3 issues, and while discussing those, take a look at the umbrella implications for all the different stages of drug development. So I want to start on the first one of the remaining 3, the third one on the list, which is surrogate efficacy endpoints that have been acknowledged by FDA as reasonably likely to predict clinical benefit. And what I'm hoping is that somebody on the panel will take 2 or 3 minutes and summarize, maybe a little more if you need it, and summarize what you heard on that point that you find likely to be most relevant to the listeners of this podcast. Doctors, KOLs, investors, execs.
I think what I heard is that in preclinical NASH, the agency is willing to accept an improvement in liver histology, as they've already defined in the guidance, and that the liver biopsy is the surrogate based on the existing research and data that we have that demonstrates that an improvement predominantly in NASH as a predictor of fibrosis or fibrosis as a predictor of clinical outcomes, histology will be the predictor of improved outcomes in patients with NASH. What we didn't hear is any other surrogate. We heard, or at least I heard, that in very early phase clinical trials, Phase 1 or early Phase 2, maybe even 2B, that surrogates could be utilized as in case ascertainment, maybe in early markers of efficacy, but in preclinical NASH, that an improvement in histology ultimately is going to be what they will stand behind as in their guidance for an improvement in clinical outcomes in NASH.
Hey, this is Stephen. So you're right, Manal. I do think there was some beating around the bush, if you will. And I use that term because the agency was very careful in their wording. I heard lots of ums as they were trying to choose the right words. Joe Turner, he's very cautious in his wording and phrasing of what's happening. Frank, a little bit more straightforward, but still they get a lot of questions. I think one of the comments Frank made was, we're getting hundreds of questions, many of those on the non-invasive testing parameters. And what they kept coming back to is, we're scientists here, we analyze data, and based on data, we make decisions. And that applies to artificial intelligence. That comment was clearly made. And then the comment about utilization of other non-invasive tests. So what I heard was, continue to collect data, continue to get data in early phase, late Phase 2B, and in 3, so that the composition of all of that data we can begin to synthesize, and you can begin to synthesize, and you can publish, and we can analyze, and hopefully we get to the point where we can use non-invasive tests as a surrogate. What I was hoping they would do, and I wrote the question and didn't get an answer to it, was, what does that look like pragmatically? We're at a surrogate endpoint that's histopathology today. Is within a year, do we think we can have AI to supplement and augment semi-quantitative assessment of the NAS components and fibrosis, and then transition to an AI endpoint, and then look at NITs based off AI data and transition then to NITs? That's what I really think is going to happen, because trying to compare an NIT to a semi-quantitative assessment is ridiculous. And I think it's going to be much easier to do that with a fully quantitative assessment of all these parameters. But I didn't hear that. I heard what I've always heard, which is collect data, show us data, we'll analyze the data. But I really think we're the ones that are going to have to collect and analyze the data and show them the roadmap to how we get this done. And I think the more we show them that, there will finally begin to be consensus. I really think that's exactly what's happened with digital pathology. I mean, how many times have we brought it forward? Now we have data. We're seeing pathologists come online with this as more and more trials are coming online. That movement has reached a crescendo to where now they say, okay. And I think that's really the path forward on a lot of these individual pragmatic steps that will help us get to an NIT endpoint. But it's gonna take time. I think based on what I heard today, we're still years away from an NIT as a surrogate.
I think the good thing about the endpoints was there's been a lot of speculation whether the FDA are moving the goalposts and whether the guidances weren't going to be able to be utilized as written for histology endpoints. And so getting that clarity today that the endpoints will remain, the endpoint is helpful for the wider community because people were concerned that there was something that was maybe going to change there. So I think the clarification that that's not changed is helpful. But I think what becomes harder to fully interpret is this sort of comment about the risk-benefit profile. And I think that's going to be mechanism of action specific compound by compound, where it's very clear that they're going to have to look at the overall approvability based on the risk-benefit profile. And even if you hit your endpoint, but they don't think maybe that the overall risk-benefit is an advantage to take forward because they're concerned about the large NASH population. And so I think that's the area where each company, each molecule is going to have to navigate through what would be acceptable.
That makes sense to me. I mean, one of the comments I noted was, I forget who it was in the Q&A. I think it was Joe Turner, in part because virtually everything that Frank and Annie said, I wound up getting only on the screen, about specifically the idea that it goes fundamentally, the same safety profile might be acceptable in a drug with a greater benefit, but not in a drug with a lesser benefit, which I think was the end coda, to me at least, on obutacolic acid that we've all been waiting to get some definition around or complete clarity around. I agree with what you say, Kitty. I think it is going to get navigated out drug by drug, but I do think that what they stated pretty clearly, at least as I heard you folks tell me, is more efficacious agents will have a wider berth on certainly side effect issues, if not major safety issues. Am I alone, or did other folks hear that as well?
Yeah, that's exactly what I heard. And really, the summary for me is the view worth the climb? Is the juice worth the squeeze? I mean, at the end of the day, if you're hitting it out of the park on efficacy, willing to accept some adverse events or mild safety issues. On the other hand, if you're marginally effective and you come with some adverse events, that's going to be looked at with scrutiny. So that was a good pickup, Roger. Thanks for bringing that up.
Thank you, Stephen. Anybody else have any, on the surrogate and efficacy endpoint, other issues, comments?
The bar is getting higher and higher for NITs and, biomarkers. 20 years ago, all you needed was a hypothesis, and you have your biomarker, you have, your disease cohort, you have your control, and you showed that there is a difference, and you publish, and that was, your biomarker. And that led to many issues, including the fact that we have, hundreds of biomarkers that are not, utilized in clinic because they really don't stand the test of time. I think we've had issues related to understanding also the effect of disease prevalence on positive predictive value, negative predictive value. I think, now you need to have your initial cohort, you need to do internal validation, external validation, multiple cohorts. It got too complicated. I think this is good, and it's good for science, and we don't want to have more biomarkers that we never utilized in real life, but the bar is just higher now, and this is, what's taking us so long. And then we have the issues with biopsy, and you're comparing to an imperfect gold standard. One thing I noticed was that they encourage having two pathologists, and I can tell you from some of the trials that we're conducting that use two pathologists, if you have them read the slides separately and score the NAFLD activity score and, give a grade for ballooning, inflammation, steatosis, and then the stage of fibrosis, and then have them compare, that they only actually agree on all of these in about 1/3 of the time. So the rest of the readings, they have to do it together and then come up with the consensus. So even having two pathologists is really not going to resolve the issues related to biopsy. The fact that even the same pathologist can read the same slide differently after a few weeks between the beginning of the trial and the end of the trial, it is, disheartening for me that we still need the biopsy, but I agree that we don't have anything to replace biopsy today.
I think I agree with both you and Stephen. I think Stephen's got good insight in the fact that the artificial intelligence platforms for the pathology are going to help us back into more sensitive NITs for performance. And on the same note, while it seems like the bar is higher, I'm not sure that it really is. And I don't know that the goalpost has shifted. I think the FDA has had its same stand. It's one thing to have a biomarker for case detection or case finding of a disease entity. And it's another thing to use such markers when balancing out risk-benefit ratios for therapeutics and looking at their performance over time. And what I heard resonate very clearly by the FDA is data. Show us the data. We're happy to weigh the data. We want you to generate the data. We want you to show us how this is pertinent for the disease, the therapeutic target, and its performance. And what I also heard is they're not going to dictate how or who we do this. They're not going to dictate who looks at pathology. They're not going to dictate how it's assessed. They're not going to tell us one pathologist, two pathologists, committee pathologists. They want to engage in dialogue. They want to see progress. But the reality is we are still in our infancy of still building data and trying to do it fast enough. And because I think of the risk-benefit ratio for a very large cohort of patients who are going to be receiving these therapeutics, we're not talking this is an orphan disease and an orphan drug. We're talking a very prevalent disease and potential large population-based safety and efficacy risk balance ratios, then I think there is some prudency being weighed.
But, speaking of data, Manal, I would like to see the data that shows that resolution of NASH leads to any improvement in outcomes. The FDA has no data. And where's the data that any therapeutic, if you improve fibrosis by one stage, that you improve outcome? It's not there yet. So I feel like, they're asking for data and they want to be scientific, but sometimes when they're pressed to make a decision and move forward with an approval path, they just take what they have. I think now they have histology and they're sticking by it. But it's really not very clear cut. What we have today is actually driven by real data. It's more like, well, we think on the initial biopsy that the fibrosis stage is the most important prognostic factor. So therefore, we think if you go from F3 to F2, it's going to be associated with improved outcome. But we still don't have that data.
I heard very similar to what everybody else heard there. The surrogate could be decided by the sponsor, but that the FDA welcomed the discussion as to what surrogate endpoints you were going to manage. And I think for me, risk-benefit ratio, I'd like to see what the threshold is. We know that the risk-benefit for having a liver biopsy and the difficulty that patients have with that and some of the outcomes of the liver biopsy and also failed liver biopsy. So I'd certainly like to see a threshold for the outcomes of trials to be certainly significantly better than the risk-benefit ratio of having a biopsy. So keeping NITs, keeping biomarkers in and strengthening the data that we're getting from them, I would like to see the sponsors discuss those heavily with the FDA.
Louise or Kitty or anybody else, but this is a quote from the UK, which is why I'm inviting you. If I get this quote too dreadfully wrong, please correct me, but I think Churchill is reputed to have said something like democracy being the worst system there is except for all the others. Does that kind of resonate? Without waiting for an answer, I'll go on and say that was what I thought these folks were saying about biopsy today. I don't think they showed any affection for it, but I think what they said was they don't have an answer they like better yet, which is why they keep asking for data, because data is the place wherein a better answer is likely to come out.
I completely agree, Roger. I do think, though, even though they said they weren't going to tell us how to analyze it, I think maybe some of the feedback I've had recently has been pretty specific. So I think they are quite good at telling you what would not be acceptable, and they are guiding companies through what would be acceptable. We recently had to submit a biopsy analysis plan where we've really detailed out how we're going to analyze our histopathology, and they want to provide feedback before we actually start. I think they're not going to tell us exactly what to do, but they definitely want to be very involved and tell you what not to do.
Sounds right. Sounds like the agency. Okay, so before we move on to the next topic, any other comments, questions, closing points anybody would like to make?
I do have two other things that I think were worth discussion. One was the histopath adjudication process. And I like the fact that it was stated that they are not defining a particular way it has to be done. They're willing to listen to what makes sense. And I think that's that's actually a step forward. They list in their white paper 2 or 3 pathologists. I don't personally like the 3 pathologist pathway. I'm more of a 2 pathologist guy, where I would like to have the slides. I would like for the 2 pathologists to come together, harmonize the readings, what they would call a stage 3. Is it one bridge, multiple bridges? How you define a classic balloon hepatocyte. What do you say is 1A versus 1B, 1C? And then they go and read independently. They come up with their own read between baseline and end of study. And where they agree, great. Where they disagree, they come together again. Again, this is the beauty of digital path. They can do that from the comfort of their own home, or if you're Pierre Bedoza, from your sailboat in the Mediterranean. And you adjudicate those differences, and whatever score you agree to is the one that you go with. And I haven't seen where that has been met with a negative reception. So there are multiple ways to skin the cat. I think we just need to get better at trying to adjudicate the issues with intra-observer variability and the placebo response rate, as Naim mentioned earlier. And I think that door is open to several different options. So I think that's maybe worthy of a discussion as well.
So I'm wondering whether that's in this section or the next one, which would be clinical endpoints for the confirmation of clinical benefit. Stephen, it sounds like you may get a lot of conversation on that one, but not necessarily in this room or in this podcast at this moment.
Well, maybe I can say one thing, Stephen. I'm completely in agreement with you that I'm in favor of 2 pathologists, but the third just seems to me that it would introduce sort of more variability, because you're going to have a third read. And so the way I had kind of interpreted that was maybe the third person is basically purely an adjudicator. They have to actually side with one reader or the other, and they become the final vote. But I certainly don't think you want a third reader involved. It doesn't seem to be that would be helpful in any regard.
Yeah, that's the way I would interpret the third reader as well. And when you poll the proverbial pathology audience for that, they all, to a person, at least to me, say, who made the third pathologist God? At which point did the third pathologist have the command authority to overrule one versus the other? And that's why I fall back to just get them in the same room, look at it, talk it over, come to a consensus, and move forward.
Makes sense. And first of all, thanks. That was a great conversation. Really, I think we covered a lot of ground and a lot of clarity. Next one to move on to would be clinical endpoints for the confirmation of clinical benefit. Who wants to summarize on that point? I'm not entirely sure I get it either, so we can skip it.
I'm not, I hope we're offline. I'm not sure that I gained much on that endpoint. I'm struggling to understand what they meant by that. One thing that I thought was interesting, and maybe, Roger, we can delve into, for the first time I heard, we've traditionally heard from the regulatory authorities about endpoints, meaningful endpoints being resolution of NASH without worsening of fibrosis, or fibrosis improvement without worsening of NASH, or hitting now both dual surrogate endpoints. But in the question and answer, I heard something very intriguing that I think lends promise. Because as clinicians, we know that if you take a pre-cirrhotic or cirrhotic NASH and just keep them stable, no change, with the anticipation that 20% could potentially decompensate in 2 to 3 years. But if you keep a cirrhotic or pre-cirrhotic from progressing, that is a meaningful endpoint. And for the first time, I heard the FDA say halt progression of cirrhosis or halt progression of fibrosis. And the halt, to me, means no change. So is it a suitable endpoint to take a stage 3 and keep them at a stage 3 with no progression? And the FDA has recognized progression to cirrhosis as being a clinically meaningful outcome. But even maybe the overarching goal of complete reversal in a time frame of 12 to 18 months is proposed in fibrosis, it may be very ambitious for fibrosis endpoints. And that halting fibrosis is the first time I've actually really heard regulatory agencies say halt, which to me resonates in stability and no change. They hinted around that a little bit, but I was intrigued with that comment of halt.
Reactions, comments?
No, I agree. I mean, I think, they made it clear that progression to cirrhosis is a valid endpoint. I was happy also to see that they said do not mix cirrhosis with NASH and, F3. As you know, previous companies tried to focus on F3, F4, and that backfired. So I think, moving forward, it's nice to keep the cirrhosis trial completely separate from the F2, F3. I think they're very clear on this point, and I hope that for future drug development that we stick to that point. I was hoping we will have more on portal hypertension and the hepatic vein pressure gradient and other potential surrogates, but I know this is also a sticky topic, and I was maybe too optimistic, but I mean, the answer we got was what we all expected. There's new data on maybe MR elastography, some MRI-based combination scores where you combine PDFF and MR elastography. We'll see, but this is all still premature.
Yeah, I'll just carry that conversation a little further. So we're kind of moving away from Manal's and following up on Naim's, but I want to just emphasize that we've come a long way with non-invasive testing. And I think we're on the precipice of having some really positive data with MRI PDFF and potentially with Pro-C3 as well. For MR elastography, I kind of had left that for dead a while back because nothing seemed to be moving the needle there. Then there was some positive data that began to come out and some even further data from Alina Allen at the Mayo Clinic. And that data is in cirrhotics. We are able to, the comment I was going to make about MR elastography that's novel and new is in the cirrhotic population, following on Naim's comment, there's a lot of momentum being built around the work that Mazen Noureddin and Alina Allen have published looking at MRE in cirrhotics predicting outcome measures. So if we're able to generate some significant data showing that regression in MR elastography actually correlates, to Manal's point, with stability of cirrhosis and preventing progression. We have historical data now to show what an MRE KPA is associated with an outcome measure. And if we're able to regress that, let's say, for instance, you have an MR elastography of 8. We know that in 3 years, based on Alina's work, 40% chance of decompensation. Whereas at 5, a KPA of 5, it's only 20%. So if we're in a clinical trial now able to take that mean KPA from 8, and let's just say for the simplistic purposes, we're really good with our drug and we're able to get it down to 5, we just prevented 50% of the cohort from progressing to decompensation. So now you can show that data to the agency. It's valid data. It's prospectively obtained data. And I think we may actually be able to move the needle quicker, Naim, on a cirrhotic endpoint than we are maybe on an F2, F3 endpoint. It's just something I want to throw out there. It's provocative and interesting. I don't know if it's got legs or not. I'm excited about what that might mean for a cirrhotic endpoint.
Speaking of cirrhotic endpoints, I want to ask you guys, I mean, how do you feel about the prevention of development of varices? As you know, this is the endpoint for some of the trials that we are conducting now. You know, you do your endoscopy at baseline, and then, you know, you're going to do your endoscopy at the end and look at the percentage of patients that develop varices. I didn't hear anything about this during the meeting. Would like to hear your thoughts.
Yeah, I think it's a tough endpoint to standardize, even amongst experienced gastroenterologists. Some of it can depend on the degree of insufflation, whether you call something a grade 1 or a grade 2. And sometimes I myself find that I may have variability in how I've graded a varix from a year or two ago to now, and I see different variabilities in grading varices in the same patient within shorter intervals based on different providers. It is subject to some interpretation variability and performance variability as well that can't be readily and equally standardized. So it's still, while it's a hard endpoint, it can't have the same issues that we have with histology, interpretation bias or sampling or performance bias.
Just picking up on Stephen's point there, we were very encouraged, certainly at the sites that we did with the hepatitis C and the cirrhosis patients, that we cured, and watching the improvement in liver stiffness quite rapidly after cure. So it does excite me that we can get into trials, certainly in the cirrhosis population. We're going to be using non-invasive markers. We're not going to be biopsying these patients. So you're correct that I would like to see a speeding up of the opportunities for those patients. And what we've also seen, and I suppose it ties in with Naim's comment, is that as the liver softens in those populations, we saw a reduction in varices and the improvement of risk benefit for those patients. So I suppose there is a combination of both of those outcomes in the cirrhotic population, and they're the patients most at need to prevent, reverse, improve, or even halt, because it improves their outcomes long term. So Manal was quite right. The halting of severe disease progression, I would say, is a really good outcome in the F3 population. Even if they didn't regress, but if they didn't progress, those benefits for patients emotionally, financially, and socially are just massive. And none of that was mentioned for patient endpoints, rather than it was all the focus on clinical. And I think we can have medication that holds a patient stable, but does improve their functionality. It might improve their cognition. So there are a lot of other endpoints that I think investigators and companies can now utilize to really look at how effective their medication is, not just on a clinical endpoint, but as a holistic endpoint for patients. And I think we have to be looking at the person as a whole rather than just one point. So I think going back to Stephen and Naim, I think absolutely the cirrhosis population, these clinical trials with some of these rapid-acting drugs could be massively life-changing in the severest form of disease.
Louise, I think that's a really interesting note on which to shift out of endpoints and into clinical trial strategies, which is that there's a whole nother level of endpoints, holistic, as you say, that wasn't touched upon directly, at least, in this webcast at all, but I think speaks compellingly to what it is we're actually trying to do to help patients at the end of the day. So I think that's great. While we've got enough time, move on to the final question, which is strategies for clinical drug development for NASH, which feels to me like it lumps together everything we've been talking about and asks, okay, what do you view as good form going forward that you might not have been as adherent to or excited about before today? And what were things that you thought might be good form up until today that feel like they might be bad form going forward?
I feel like, it's the same points we made earlier, that we have another endpoint, which is both NASH resolution, fibrosis improvement, the fact that cirrhosis should be separated. We talked about some of the issues related to cirrhosis endpoints being clinical at this point, the lack of biomarker strategy that we can rely on for drug approval.
Okay, so let me toss out a couple of questions. If we're now more clearly defined on the importance of risk-benefit and the idea that acceptance of your safety profile is going to have to do with your efficacy profile, how confident do you need to be about your efficacy advantage versus other products that are further in development to decide to go ahead with what might wind up being a larger phase 3 trial than you originally had the appetite for? Do you folks see that as a legit question, or am I misinterpreting what I'm hearing today?
I'll take that, and I'll say this. So I think my colleagues would agree with me that combination therapy is still ultimately going to be the solution for a significant majority of patients with NASH and fibrosis. And if that is the case, we need to step back and realize that every drug moving forward doesn't have to jump a higher hurdle to cross the finish line, because we don't know what synergy looks like necessarily. We have elegant work that Naim is working on and others looking at some of these early combinations. But, and I hate to say this, Naim, and I don't mean to offend anybody here, but I don't think that the best combinations have been put forward yet so far. I think what we've done is we've done combination by convenience rather than combination based on sound scientific rationale. And as Scott Friedman will always say, gene analysis and looking at which genes are upregulated, downregulated when put together in a petri dish and then in a preclinical model and taking that forward. I'm not suggesting that we have to do it that way. I do think that if you have a drug that is having a positive histopathologic benefit and potentially additive extrahepatic benefits that maybe don't quite meet the hurdle of another drug drug that's in front of it from a histopathologic perspective. It does not mean that drug can't be used in combination. And I'm thinking of several of these. Let's just say resmetirom crosses the finish line in 2023, and it may or may not be the first drug to treat NASH. Well, we know that it's oral, it's well tolerated, and it has some lipid effects in a positive way, but it doesn't mean that it can't improve when combined with another agent, whether injectable or whether it's another oral. So I think I don't want to be short-sighted in that regard. The other thing, before I forget about it, that to your maybe your earlier question, and this is, I would love Kitty's input on this as well, since efruxifermin, at least in the phase 2A trial, where there were liver biopsies performed, seemed to have a very robust histopathologic response in a very short period of time. The comment that the FDA made around 12 to 18 months for a phase 2 trial, looking at histopathology, I think is reflective of what has come before them so far, which are drugs that, for all intent and purposes, were not incredibly potent, right? So we've seen beta-cholic acid come across, we've seen elafibranor come across, and others, but the needle wasn't being moved in a huge direction histopathologically. And so the framework, the mindset is one of we need to treat longer to show a positive benefit. But some of these newer drugs coming behind are I think, moving the needle in a much quicker fashion. And so do we really need a 12 or 18-month phase 2B histopathology study to inform our phase 3 data? I throw that out there as a question because they kept coming back to that. There was a question about it in the white paper. It was 24 months, 2 years, and then they kind of backed down to 12 to 18 in the presentation. And I just thought that was interesting.
So, Stephen, I completely agree with you. And I do think that the guidance has got to be written in a way that it is sort of appropriate for various different mechanisms of action. And so I think the agency are quite conservative here. And it may be, maybe to Louise's point earlier, looking at more indirect mechanisms that are more indirect in terms of antifibrotic. And like if we use hepatitis C as a surrogate there, when you remove the virus and you allow the liver to then have time to repair, you do get that sort of indirect effect on being antifibrotic, but it does take a long time. And I think there's outcome studies that, really you're talking sort of 2 to 5 years to see that indirect method of being antifibrotic. I think, some of the newer generation mechanisms are really looking at being both metabolic. If you can remove the fat, I think it's clear that is affecting your steatosis score and then hopefully your NASH resolution. But if you can also then be directly anti-fibrotic, and I think the data we have with the FGF21 and EFA is suggesting this, that we actually are seeing direct antifibrotic. And if you can see that, you can see it early. But the combination of those two together seem to be suggesting that we could look at shorter timeframes in terms of these efficacy endpoints. I think that the one thing we have to do is we've got to create the data. So we're actively moving that forward. And, it does seem that you can negotiate those timeframes with the agency. And I think, the one thing I'm always sort of listening to is I think they kept saying, be careful about just powering and focus on efficacy, and you need to be taking safety into consideration. But if you continue your patients onto long-term follow-ups and you're continuing to generate that safety data, I think that might be something that you can do to kind of sort of alleviate that concern. And it may be that you just provide later data cut at certain points for your safety and combine that with earlier cuts for your efficacy. But there's ways to navigate through that.
I think one important point that Joe Turner made when he was asked about what is a clinically meaningful effect size, he said anything better than placebo. I think this is very important. He made the point that they're going to take safety into consideration and then decide. I think this leaves it vague, really, and the FDA can basically do whatever in terms of that risk-benefit ratio. So you can hit the efficacy endpoint, but they say, well, the safety profile doesn't justify it, but how do you do this? I think the way I look at new drugs, you have your liver similar endpoints in terms of fibrosis, NASH resolution. You have your metabolic syndrome endpoints in terms of weight loss, effect on A1C, insulin sensitivity, dyslipidemia. And then you have, your adverse events of interest, whether it's GI side effects or weight gain, or it depends on the drug. So, you're going to put all of these in one formula and then decide if the risk-benefit ratio justifies approval. But it's not very clear, how much weight you're going to put on each of these. I couldn't agree more with Stephen that, I think we're moving toward a future that looks like the management of type 2 diabetes. It's not going to be just one drug that fits all. In diabetes, we have sometimes, better drugs that, will help in terms of efficacy more, but they're not tolerated well. And many diabetic patients still take older drugs that may not have the same cardiovascular benefit that we see with some of the newer drugs. But I can see a similar paradigm for NASH.
I agree fully. I don't think it's going to be one drug fits all. I do agree that we're going to be looking at combination therapies, but one of our limitations from going in that direction is we have not yet defined the abundancy of heterogeneity in these cohorts. We throw one drug at them, hoping that it'll work, but there are responders and non-responders in every trial. So we haven't phenotyped these patients or genotyped these patients accordingly to be able to optimize any unique therapeutic for the most optimal, safe and efficacy outcome that we're hoping to achieve. And we have learned lessons. There are ample anti-glucose lowering medications on the market, not one size fits all for any of our diabetics. And we've learned lessons from our hepatitis C era, where we initially treated all hep C like it was the same disease, only to discover that there were unique genotypes, and then we're able to tailor therapies, therapeutic durations, and therapeutic approaches according to the genotype of a virus. And I think we really need to genotype our own patients to understand what their unique pathogenic drivers for their NASH and NASH fibrosis is, and tailor therapeutics accordingly. In which case some patients may receive single therapy and others combination therapies, depending on typing our patients accordingly. But overall, I think we are going to be moving to combination therapies.
So, Manal, that entire comment makes a ton of sense to me on 2 or 3 levels. Number one, to get that done in diabetes took multiple classes of drugs that got approved as monotherapies and a lot of years and a fair amount of failure, or at least what I'm going to call marginal success. In that regard, Stephen, I want to go back to your comment about combination therapies. Do we envision that an agent that could not get approved in monotherapy would go all the way through development and be approved as part of a combination therapy? Or do we think that even if it's not a great drug, it's got to get approved in monotherapy to find its way in a combination? Question first to Stephen, but then to the group.
That's maybe $1 billion question. What does it take to get a drug into clinical practice if it's going to be a combination agent and not a standalone agent? Maybe another way to ask the question. And I'm not sure we have that answer because of the cost involved. I'm not a pharmaceutical company. Maybe Kitty's better to answer that one. But if I were, I guess I would look at my asset and say, well, what's the cost of developing this relative to the opportunity that exists for this drug in the market? And if I did my phase 2B, got histopathology and compared it, with other drugs ahead of it, then I would have to think about what are my opportunities to partner with somebody that has an asset that could be used in combination, or if I have an asset in-house, could I combine it with that and move it forward versus moving it forward on its own merits, showing that it's efficacious, but it's being inferior to something that's already on the market. I don't have a good answer there. I think it would really depend on a lot of different dynamics, only one of which is the drug's efficacy.
So, Kitty, what do you think? Which route makes more sense to you?
Well, maybe I'm a little biased or maybe the eternal optimist, but, I still have hope for monotherapy. And I honestly think that if you could just have one drug, it would obviously be the simplest option. I do think it does get complicated when you start combining mechanisms of action. And I think some of the combinations that have been put forward are trying to balance out sort of a liability with one drug with another, which, you kind of end up with a net zero. Like, you maybe have a triglyceride increase and the other drug will decrease it, but overall, these patients have very high levels of triglycerides. And what you don't want to do is just take them back to baseline. You would like to improve overall. But I think that combination therapy has been used in multiple different therapeutic areas. Coming from anti-infectives, whether it be HIV or Hep C, where, you've seen very nice partnerships. Companies will come together. And I think if you can come up with the right combination of drugs that affect the overall comorbidities of NASH, as well as the NASH itself, I think it would always be something positive to take forward. And I think companies, there's a lot of combination studies going forward right now where, we are looking at drugs that complement each other. It just is a more sort of difficult drug development program. You've got more people in the partnership. For the patient, there's a negative. You've got to co-pays and the costs. So, I think it all depends on whether there's a potential monotherapy that could go forward. That would be my first choice. But if not, you have to then work out, as Stephen says, it's the right combinations that really are overall treating the NASH, but also the comorbidities, in my opinion.
That sounds like most of the clients I've spoken to in this or other disease categories in terms of preference for monotherapies. A, it's easier to build a case, and B, it's easier to get it done. I'm trying right now to think of drug classes I know of where agents that did not get to market as monotherapy became successful as part of a blockbuster or significant combination. I'm stretching. I don't have one off the top of my mind. We're about 10 minutes from wrapping up. In concluding, I have just a couple of questions I'd like to know about. Number one, and maybe I'll do number one and we'll go around the table, and then number two and we'll go around, number three and we'll go around. Number one, particularly after having processed this for the last hour, what's the biggest news item for you that emerged from this webcast? The biggest new thing or the biggest important thing?
I think for me, it's the 2 points that I made earlier. Number one is we have a new endpoint, which is both NASH resolution and fibrosis improvement. The fact that we have potentially a new design for phase 3, where you start with the cirrhosis trial, and that will be an outcome trial, and then the F2, F3, and that will be a histology-based trial. I think these were the two main findings for me that got me excited, that may actually change a little bit how we think about designing a new clinical trial.
Okay, next.
So this is Kitty. I agree with Naim. Those were, I think, the two main. I think for me, just to add on to that, I think just that clarity around the use of digital pathology was very helpful. I know it's just a very practical implementation, but I think, being able to digitize those images and the potential opportunity to use the artificial intelligence will be helpful for the field.
Well, I don't know that this is a news item, as it is a message that resonated with me. And clearly the FDA is excited about this area of work, is wanting to partner, and interested in dialogue, wanting to help propel the discovery of biomarkers forward. And they want to see histology go away as much as we do. And so I think I was excited to see the openness about exploring such biomarkers, but the onus of responsibility is on us to generate the data. To allow them to deliberate it thoughtfully.
Two things for me really struck home. Number one, I'm so thankful that Kitty asked the question about digital pathology, because I get asked that every week from sponsors. Can we move to digital path? And now we can say, yes, we can move to digital path. That was huge. Number two, I think what has become clear through communications from the FDA, to include former communications, the white paper, the webinar today, is that safety is half of the equation. These patients, NASH patients, are at risk of cardiovascular death and all the complications that go with that and all the factors that drive that. And if you have a drug that is at risk of worsening that, they made it clear today that the hurdle will be significantly higher. And it's not just that risk, it's other adverse events as well, particularly DILI-related risk. And I think that became clear today, that you have to have a clear DILI algorithm for your study to adjudicate results related to a potential DILI outcome or a concern. So I think, and there's a lot of other areas, I think, where maybe it wasn't as clear, but we had some positive signals, and I think that dialogue will continue to evolve. And what they say today with additional data may change by the time we get halfway through the year, and I expect it to, right, as the field evolves. So I'm walking away today, again, a 5 out of 10 is what I said at the beginning. I still think that's the case. There were some positive things that I'm walking away from, and some things that I think we need more work to do to provide them data to analyze to give us what we want to hear.
Okay, thanks, Stephen. Louise, biggest story out of today for you?
I think for me, it's obviously the strength that they're asking for us to put onto non-invasive techniques and biomarkers to take us beyond the biopsy if we can over the next few years. And I would hope that the timing of this allows for all of those protocols that are currently on hold or have been suspended due to COVID and the difficulties with that, for some of those protocols to now include more non-invasive technologies to be able to look at better outcomes in addition to what they were probably doing before. So I think it strengthens that for me.
Okay, I'm going to say two things real quickly. Number one is the degree to which I think the agency would like to move on from histology once it has the data to do that, although it won't produce the data. The academia and the industry have to, but they clearly want to do that. And then second was the importance of safety in the equation. And Stephen, thanks for mentioning DILI, because that jumped out at me and I'd not mentioned it yet. Second question, try to do this in one sentence or less. The most surprising thing you heard today?
The combination idea of taking parallel well-compensated cirrhotics and then the standard F2, F3s and running a phase 3 parallel trial to get surrogate endpoint approval, but then no need to continue in a phase 4 program, just follow out your cirrhotic cohort to an endpoint. There's a lot of questions around that, right? Because what happens if the cirrhotic trial doesn't show a positive impact, but yet you have Subpart H approval? So, I mean, there's other questions I think that we have to ask about that, but it's intriguing that that was brought forth by the agency as a potential track for a phase 3 program.
I thought the clarity around the idea that goes, even if you've hit your efficacy mile point, if you're not safe enough, it doesn't go forward, and the story that told about what we've all been wondering since June of 2020. I thought that was not surprising, but the clarity around it I thought was fairly, it's clearer than an agency usually gets.
I'll pass on this one. No big surprises for me.
Yeah, I think Stephen said it really is the big surprise.
Third one, no one pass on this one, please. The thing you heard today that will have the greatest bearing on how you perform in NASH development related portion of your work over the next 3 to 6 months.
I think how I perform NASH related research, and especially as I deliberate issues related to study design, is more of a close eye on early anticipation and deliberation and projective planning with adjudication, with algorithms to safeguard against risk, or how algorithms should otherwise be managed in clinical practice, whether it be proactive management of a lipid signal or very detailed approach to liver biochemistry alterations. I think we have to anticipate potential drug-related side effects and plan for them with algorithms in the conduct of running our clinical trials to minimize risk or manage it, such that when data comes to FDA, the anticipation of safety was there, the mitigation of safety was there, because clearly they're weighing both equitably, and it would be a shame to see safety issues that would have otherwise could have been readily managed to mitigate safety, not done so before FDA sees a compilation of data. All of this certainly is hindsight, but I think as we plan our protocols and clinical trials, we should be defining algorithms for safety management, whether it be glucose or lipids or weight or liver enzymes, such that we are well informed as to what investigators and sites do and don't do. And because there's so many different approaches to managing the driver of the disease, we got to remember that NASH is an endpoint, not an outcome of other diseases. But because there's so much variability with how physicians manage diabetes and lipids and blood pressure and weight, we almost need to systematize these approaches in the context of our protocols such that we aren't caught off guard in our ability to interpret the outcomes.
Okay, thanks, Manal. Stephen, I know you've got to drop at the top of the hour. Why don't you go next?
What I'm taking away from this is that digital pathology can be used, and that there is not a hard written in stone adjudication pathway for pathology interpretation, which I think is great, and the fact that they're really interested in artificial intelligence and how can we bring to bear fully quantitative assessment. And I will continue to encourage and push for AI anytime we're getting liver biopsies, because I think that is really the next giant leap forward in NASH drug development.
Okay, thank you, Stephen. Naim, Kitty, Louise.
I think for me, we heard clear that the FDA needs more data to qualify biomarkers as endpoints. And my goal over the next 3 months or so is to start working on, studies to generate that data. And my bias, I'm more excited now about combination biomarkers. So instead of just looking at PDFF for steatosis or PRO-C3 for fibrosis, I'm more interested in biomarkers like the FAST score, where you include the CAP score for steatosis, the liver stiffness for fibrosis, and the AST for inflammation, and look at them as just one endpoint. We've presented some data at AASLD looking at the Gilead combination therapy and the effect on the FAST. Intercept presented some data also with obeticholic acid. There's similar data being generated now with the MRI parameters where you combine MRE, PDFF, AST, or potentially other fibrosis biomarkers. In my mind, probably this is the future also. So it's going to be the combo approach, combination biomarkers as one score, and then maybe combination therapy. So I think this is, the FDA made it clear, generate data and we will review it. And I want to be part of generating that data in my clinic and conducting these studies. One other thought I had was, if you're a company today that you have, one big trial for F2, F3, and you're going to do the phase 4, would it make sense to also design a cirrhotic trial independent? And then you have 2 chances at hitting that outcome endpoint. I don't know how the FDA is going to look at it. I mean, would you be allowed to have 2 chances at hitting outcomes? Just a thought. And then one final thing is I wanted to just get back to what Manal said about genotyping. I think this is the future. We are seeing more of this RNA interference now, and we can design these short interfering RNAs, ASOs, to target, for example, the PNPLA3. I think we're going to get into more precision medicine, and I think if you're going to do combination therapy, probably this would be the way to do it, where you select the appropriate patient for the appropriate therapy.
Thanks, Naim. Kitty, you want to go next?
Yeah, I think in the immediate future, I think we're operationalizing our histopathology analysis in a way that I think is in alignment with the agency. So that will be something we'll be moving forward with quickly. And then I think we do have that goal of coming up with the most accelerated drug development pathway and sort of looking at some of the suggestions the agency moved today about how to use your clinical outcomes in F4 along with your F2, F3 patients on the surrogate endpoint. So trying to work out, how that would really work. So I think that's where we are. And then Naim, we are working on some interesting correlations with our data for EASL. And I would love to talk to you more about what you think about are the right combinations, because I do think that's what we're going to have to look at. Is it's not just one biomarker, it's combinations.
I would love to look at the data. Yes, please do.
And Kitty, thanks for a really superb first contribution. It's been great having you with us today. Louise?
I suppose my goals were probably less than the others in the context that I obviously provide a service that tries to provide the highest quality FibroScans. So from non-invasive technology, it's about the accuracy of FibroScan. Everybody says it's a relatively easy test to do, but it's also a very easy test to get wrong. You only need a couple of A or E waves in a small proof of concept study or phase 2A study to actually skew your entire data. So to be able to use FAST and FibroScan just in general, it has to be very accurate. So it for me, it's about the quality and maintaining that quality and effectivity. And we do a number of FibroScans, so it's not one user just doing it for a small cohort of patients and then not doing it for another 12 months. And so for me, it's about focusing on that data. And as Naim rightly said, that opportunity to be able to use that and provide that to clinical trials with the highest quality is key. because it is about a patient outcome in the end, and proving the proof of concept is vitally important. And then also getting into the larger studies. So it will be all about the efficiency and the effectivity of those tests for me.
Thank you, Louise. Okay. And from my perspective, I think what I've learned is that progress is more likely on a couple of fronts, but may take longer than people had hoped. That we're going to be with histology longer, which I think will slow up the process of getting trials done. People might need longer trials or trials with more patients in them or creative strategies to adjust, and all that may take time. But that the agency seems to be looking at a lot of the same challenges with how things are done now that we are, and are looking for data and ways to make it better. So I believe over time, things will get a lot clearer. It just may take a little longer. With that, let's wrap up for today. I want to thank a fantastic group of panelists, Naim, Louise, Manal, Stephen, and Kitty's first effort, which I said I thought was really exceptional. I want to thank my team, Mike Wilson, who will be spending the next few hours trying to turn this into something we can produce quickly. Eric Rounds, who will be promoting it. Palatea Lee, who makes sure that everyone's got what they need to sound good. And you folks who listen to it. This episode will launch in pieces over the weekend. Over the next week, we will be putting out different portions of this and the pre-webcast. Maybe not a new episode on Thursday. We're still debating that. The next new episode I know we're doing will launch on late the evening of February 10th, the morning of February 11th, summarizing the recent British Medical Journal review article on treatment of NASH, of NAFLD. And with that, thanks everybody. Thanks for staying with us. I hope you've enjoyed this. Please send comments. We will also be announcing our new music when we come back on February 11th. So good luck to everybody all week. And if we're not on next week, stay safe. Surf on. We'll see you on the podcast. Bye-bye now.
You've been listening to the Surfing the NASH Tsunami podcast. Have any questions for the surfers? You can send them to surfingnash.com and we will answer on the podcast or the website. Thanks for listening. See you next week on the podcast.